All right, guys, I must care about you a lot because I hate filming myself. I want to show you the difference between meiosis and mitosis. Right? This is mitosis. We've already talked about this. Mitosis is where you start with a single parent cell and you end with two identical daughter cells. Right? The daughter cells are identical to each other and to the parent. This process is used for repairing injury, for growth, so going from a little organism to a big organism, and for replacing dead cells. Notice in mitosis, you start with your cell, it's a diploid cell, that means there's pairs. This is one homologous pair, this is another homologous pair. These guys double, so they replicate themselves, and you end up with these sister chromatids. So this is still a homologous pair, but now you have your original strand and a copy, original strand and a copy, right? These guys have also doubled, so now you have two homologous pairs of sister chromatids, right? Then what happens during mitosis? These guys split apart. One of these two chromatids goes here, one of these two chromatids goes here, and that happens with all four. So you end up with two diploid cells, daughter cells. Diploid just means that they're homologous pairs, so pairs and pairs, right? That's mitosis. Now we're going to look at meiosis, or some people say meiosis. Meiosis, the point of this is to make human gametes. Gametes are sex cells, and they're haploid. That means there's no pairs. That's really important because when you have a sperm cell and an egg cell that come together, they combine their chromosomes. So if you were to have 46 chromosomes in your sperm cell and 46 chromosomes in your egg cell, if they came together, you'd have 92. That would be a problem. So you need to have half as many, 23 here and 23 here. When they come together, they make 46. It's also important that you have half of each pair. So you'll have half of one homologous pair here, half of one homologous pair here. When they come together, there you've got your pairs, all right? So you have a haploid cell, they come together to make a diploid. So let's look at how meiosis works. You start just like you did in mitosis. You start with a diploid cell, this is a body cell, where you have two homologous pairs, right? Then we double our chromosomes, like we did before, or double our DNA. So now you have two pairs of homologous sister chromatids. Now what happens, it's kind of weird. So you have your strands here, your homologous chromosomes, your chromatids. What happens, they get really close together, and when they do, they swap some genetic material. So notice how this one was all green and this was all blue. They get really close together, and a little piece of this breaks off, a little piece of this breaks off, and they swap places. That's really important because what you're doing is you're creating new combinations of chromosomes now that are unique, that have never been seen before. So now we still have our homologous pairs, but they're a little bit scrambled, right? So here's one and here's one. You know what I forgot to do? I forgot to add my little piece right here. There we go. All right. Now we have our first division. So now these chromosomes are going to split, but unlike it did in mitosis where they went like this, what happens here is they go like this. So this homologous pair gets split apart. So they're no longer a pair. So this one goes down here, this one goes down here. So now we are a haploid cell. This is a cell with no pairs of chromosomes anymore. All right, same thing here. This one went here, this one went here. So now we have basically half as much DNA as we had before, half the information. Now it's going to divide again. So now it looks more like mitosis, where this guy goes like this guy goes like this. So now we're haploid still, but we're haploid with no sister chromatids, all right? So now let's compare our daughter cells. Look at the daughter cell here. We're diploid. We have one chromosome in each pair. Here we do not. We only have half of that first pair. Half of it, half of it, half of it. So we have now 4 chromatids, half of each pair in each of our 4 daughter cells. And notice how they're all different. This doesn't have any green on it. This has green and blue. This has blue and green, and this one is green. Same thing with the little guys. Here we had a pair and a pair. No pairs here, all right? So we have a red and blue, a red and black one, just a red one, just a black one, and a black and red one. So now we have 4 unique, non-identical daughter cells, and all of these guys are going to be haploid gametes.